All right, so good evening. Thank you all for joining us tonight. Today is our soil health call happening every other month, the first Monday of the month. And today's topic is going to be around seeds and soil health. The reason why we chose this topic is because this is the time of the year where we're actually getting very excited about the possibility for, just getting our seeds going for the garden. So we're going to go over some principles and some how-tos. We'll wrap that up quickly, and then we'll head in directly into discussions. And then we'll pause in between as well as we go through the modules here. All right, great. So let's see if I can advance my slides. All right, so as I mentioned, there's going to be three major topics that we're going to be covering tonight. These are going to be the background on conventional seed cultivation, uh, importance of seed saving for soil health, and why is it important to inoculate your seeds? Just bear with me a second. I'm having some issues with my slides here, of course. Oh boy, just 1 second, I'm sorry. I had some notes. Oopsie. Not sure what's going on here. My notes are not showing up on my slide.
You have a show and presenter view?
Yeah, it's some presenter view.
Hmm.
Not sure.
Do you want to send me the slides and I do the slides and you have it in your computer?
Um, this is a PowerPoint, so I would probably have to upload it.
Hmm.
I'm sorry,
Oh, yeah.
guys. Just give me 1 second. I'll just stop the share for a second. Thank you. Let's try this again.
You can do it in this view. That'll work. You know that, right?
Yeah, let me just see if I can. Oh, my mouse disappeared too. Let's see
If
if this.
all else fails, you can use this view.
All right, let's do it that way. Yeah, let's do it that way. Thanks for your patience, all. Um, all right. So again, these are the topics that we're going to be discussing. So let's get right into the background of this. Um, so conventional seed cultivation essentially refers to the traditional farming practices that have evolved over centuries. These play a crucial role in the development of modern agriculture and food production systems. And understanding this historical context is vital for appreciating the roots of our current agricultural processes and practices. So let's go ahead and dive into some of these aspects of this conventional seed saving. So as many of you know, the Green Revolution initiated around the 20th century, and it was a fairly transformative period in agriculture. It introduced highly yielding varieties, chemical fertilizers, and pesticides to boost crop production. And it also significantly increased global food production, addressing hunger and malnutrition, although there's some clear aspects of that that are in contention, and we'll talk about these a little bit here. So what are these key practices that we find in conventional seed cultivation? You can see here we actually have 5 topics that we can go over quickly here. Monocultures do exist in conventional seed saving, where a single crop in a specific area is grown over time. And while it simplifies management, it can lead to loss of biodiversity and increased susceptibility to pests and diseases. And in order to combat this, crop rotation is a common practice to remediate some of these issues that are found in this type of practice. Hybridization is another aspect of this too, of seed saving. And it's the crossbreeding of different plant varieties to produce hybrid seeds. These hybrid seeds also exhibit desirable traits such as increased yield and resistance to diseases. The issue with this is that, farmers have to purchase seeds year after year, contributing to the dependency on seed suppliers. Next, we actually have GMOs, genetically modified seeds, and these essentially are just that. They alter the genetic makeup of crops and introduce specific traits. They can confer resistance to pests, disease, and tolerance to environmental conditions. But again, has sparked many debates about safety and the environmental impact and intellectual property rights as well. Chemical pesticides, fertilizers are widely used in conventional agriculture to control pests and enhance soil fertility. And while these increasing yields can lead to environmental pollution, soil degradation, and harm to non-target organisms as well. Regenerative practices or alternatives and integrating pest management are some of these management practices that are used to somewhat combat the excessive use of these pesticides and fertilizers. Seed patented or patenting also involves granting intellectual property rights to seed varieties. Corporations patent genetically modified seeds, impacting farmers' ability to save seeds and traditional farming practices as well. This also raises questions about the genetic resources and the lack thereof of the biodiversity. Monoculture, we briefly discussed this already. Again, contributing to the loss of biodiversity. Susceptibility to crop failure due to pest, disease, and environmental changes. Genetic uniformity is another thing that we see, and these are some of the criticisms, right, that we can take apart from this conventional seed saving. And there's also a limited number of crop varieties when you actually have this uniformity in genetic processes. Again, and this is something that we'll discuss again and again and again, it's the increase of diseases and pests during these practices as well. Again, diverse genetic backgrounds provide resilience and adaptability to changing environmental conditions, and the excessive use of fertilizers and pesticides also contribute to the environmental pollution, such as eutrophication and contamination of our bodies of water. Soil degradation, again, water contamination and harm to non-target organisms are consequences of these chemical inputs. And also, we'll also discuss a few more other impacts that these also have in a few more slides ahead too. And finally, there's also a heavy resilience on these chemical inputs creates dependencies amongst farmers that unfortunately burden them uh, with the socioeconomic impacts that these have, ultimately reducing their ability to receive or achieve sovereignty on the farm. Okay. I'm going to pause there for a second and make sure there are no questions before we proceed to the next set of slides. And again, we're going to do this in less than 30 minutes, so we can have time for discussions on specific topics that we have covered here. All right, so I don't see anything, so we shall continue. So right now we're going to discuss a few aspects of why is it important to save your seeds for soil health and how saving seeds relates to soil health. But before we begin, we're going to have to talk about a few key concepts here. And these are endophytes, specifically related to this thing called the rhizophagy cycle. I'm not going to go into much detail about this. There's a lot of information out there on this rhizophagy cycle. You can just do a quick YouTube search, and you'll find a ton of information on this cycle. But briefly speaking about this, about the role of endophytes, which are just organisms that reside inside of other organisms without any detrimental impact. We have the cycle where we now understand that plants have been adapted or have prefer to get their nutrition in the wild with the aid of bacteria and other organisms. The mechanism essentially works as the plant releases exudates, other sugars, proteins, acids, a biofactory of chemicals are released into the soil. These attract specific microbes. The microbes are then actually taken up by the plant roots. Once inside the plant, there's a process called cyclosis where the plant sends it almost like a washing machine, if you will, and sends all these organisms and sends a superoxide chemical that breaks down their cell walls. And so if the bacteria or whatever microorganism is that the plant consumed was consuming a specific diet, then let's say for phosphorus for them, for this example here, let's say the organism was consuming phosphorus. Once inside the plant can now take up that phosphorus. The beauty of all this is that it is a cycle, and so when the plant has received what it was obtaining from that microbe, it then actually starts to expel it out. But before it expels the microbes out, and this is actually what causes root elongation as well. But before it actually sends it out, it gives it all of the necessary parts. So if the microbe came in with some flagella, it'll give it all of its needs to rebuild itself so that the process can continue happening. Now, this is important because some of the key concepts that we're going to be discussing now will be important for what we're trying to achieve here when it comes to soil health and endophytes. So again, some of these benefits are listed here. There are many, and this is a fairly new discovery. It was discovered in about 2010, actually, as not as a cycle, but in 2010 it was discovered that the plants actually are taking these microbes up. But in 2019 it was discovered that it was a cycle, and so there's still a lot of research going on in the area of endophytes. Endophytes have been studied for many, many years, but for our knowledge, the most common endophytes that we know of, or we knew of before this rhizophagy cycle, were fungal endophytes. So a lot of times we know that grasses associate with certain fungi. We also have mycorrhizal fungi, endo and ecto. And so these are some endophytes that we know of. But this is, again, a new technology or a new discovery. So again, better plant development and growth, more nutrients into the plants. It's been discovered that that endophytes can supply up to 80% of the plant's nutrition, which is somewhat incredible that they can do this, which is why we actually see plants doing pretty well in the wild without human intervention. This is how plants were evolved to receive nutrients. Um, Resistance to diseases, again, once you actually have a complete biodiversity in your soil. Uh, the idea is that you would actually also see these inside of your plants, causing pathogens to be less virulent because there's competition inside the plants. Plants are hardier and resistant to stress. There's some endophytes that actually protect plants against cold, against abiotic and biotic stressors, so cold, heat, halophytic, meaning high tolerances to salt, and the list goes on, actually. They also have the ability to inhibit competitor plants, and some of the research that has been done around this, I believe they used some type of Phragmites, where they took some of the endophytes from the Phragmites and put it into the weeds to suppress some of the weeds. So this is something that's being studied and looked at, and really cool if you ask me. And also, the endophytes may deter animals and insects from feeding on plants, and this is probably because the plant is just producing less simple sugars if you have a complete microbiome in the plant. So we're going to quickly go over some of the seed treatments that hurt endophytes. We now know what the endophytes are, their benefits. So I want to quickly go over some of these practices that are done now, whether that's conventional or even in your backyard. It's important to understand some of these things that reduce the amount of endophytes. Now, I'm not saying that we should not do one thing or another. It's just important to understand some of these principles as to how they relate to how the plant itself gets its nutrition. So one of the things here are the removal of the seed husks. This can result in the seeds acquiring antifungal treatment, which is very, very common in the industry, in order for the seedlings to fight off pathogenic fungi. Sterilization of seed surfaces. This is, again, very common. You see this common practice. Acids like TSP that are typically used to increase germination will destroy the endophytes that are on the seed surface. And this goes to any acid too. Any weak acid could have this detrimental effect. Failure to let seeds mature in the field. So this is actually a very interesting concept because these endophytes, the main purpose of these plants are to reproduce, right? And so if their main purpose is to reproduce, they're spending a lot of energy trying to make sure that these endophytes do get to the reproductive parts, meaning the seed ultimately. And so while the plant is supplying all of these good endophytes that it used to grow optimally, um, the uh, these microbes are then actually found in the next generation of seeds. However, other microbes do colonize those seeds as well as the plant goes through this maturation phase. So picking them too early may have detrimental effects when saving seeds, as um, a complete microbiome in terms of endophytes will probably not be present there. So, waiting a little longer and trying to mimic some of these natural processes in the wild will perhaps give you a wider range of endophytes. Coating seeds with fungicides, again, highly detrimental. This is obviously going to have negative impact on any uh, fungi that may actually be beneficial to your plants. And so again, they also reduce the amount of endophytes present. So I wanted to bring up this concept of dysbiosis. And essentially, this is something that, and the reason why I want to bring this up is because we're in this interesting feedback loop where both plants and humans have this symbiotic relationship, and one cannot exist without the other, right? So if we now know that these endophytes are actually very important for their plant microbiome because it protects them from biotic, abiotic stressors, it's bringing the nutrition that the plant needs, um, meaning that we probably actually can achieve higher nutrient-dense foods. These same endophytes will actually be the ones that we consume as well, and are also part of our microbiome. So if the plants don't have these, it'll be very difficult for us to actually also receive some of these endophytes as well, and some of these beneficial microbes that are present all around the plant. And so I want to bring this up only because, we are not very dissimilar to plants. Um, and so making sure that we're actually getting all of these endophytes properly into the plants will assure that we also get that nutrition as well. So quickly here, we're going to go, this is going to be the last step here. I will go quickly here on seed inoculants. And so we already discussed all the benefits of these endophytes. And you may be asking yourself, well, why should I promote endophytes? And the answer is very clear, right? We want to make sure that our plants are healthy, that they're photosynthesizing to their max genetic potential, and that they're actually producing nutrient-dense foods, which is the ultimate goal for us as cultivators. Um, and so there's a lot of confusion behind what seed inoculants are. And there's a lot of talk right now amongst many researchers on the fact of, do we get microbes, or is it uh, these things called inducers, meaning is it the microbe itself that we want to be inoculating the seed with, or is it just like a cold water extract of, let's say, a Johnson-Su bioreactor because it has all of these autoinducers, which are essentially hormone-like chemicals that all microbes actually use to sense their environment. And so if we actually, so there's a debate between these two things, do I need to be putting microbes, or do I need these autoinducers? Um, I don't have a stake in one or the other. I think that there's no harm in trying to get microbes onto the seeds. And the reason why I say this is because if you're not growing your seeds in a highly microbially rich environment, more than likely you won't have all the endophytes that you need. And so as a cultivator, we need to ensure that these endophytes are actually getting back onto the seeds. Also, if we are purchasing seeds from commercial varieties, endophytes are less likely to be on there. And ultimately, there's also adaptability to your site. So if you are saving seeds from your own environment, these endophytes have been chosen by the plant to give certain properties. So if you're purchasing a seed that came from, let's say, Georgia, uh, those endophytes will be very different from the endophytes that you will need up here. And so inoculating is probably something that should be standard practice no matter where you're getting your seed from, because there's that adaptability there. And in terms of seed inoculants too, there's, we're looking at, again, mycorrhizal fungi, we're looking at nitrogen fixers and a combination of both. There's a myriad of these seed inoculants in the market. Um, and I think that having ways to test whether or not these inoculants are working for your specific context would be the way to go. But again, we should be doing it. Um, and now I'm going to actually stop my share and open this to questions now. Questions, concerns, anything that comes to mind? Any clarification needed for any of the things that we talked about? Specific questions that anybody has about their own seeds and what inoculums are using, and should you be using a certain inoculum? I also would be interested to see if anybody received my email earlier. Um, there's some really, really good resources in that email. There's specifically three. One is I believe she's a microbiologist, but and I gave a timestamp. She talks about these autoinducers. And although she's speaking about the human body, these are all in the same, we're not different from, sorry, the microbes don't behave any differently. So for example, in there, she talks about how these hormone-like compounds are sending signals to other uh, microbes to create what's called quorum sensing. So they won't behave in a single fashion. Rather, they would actually behave in a way where they can all signal each other all at the same time and say, "Hey, we have a cousin in here, or we have something that's unrelated to us. Let's express our genes in this way." So this has a lot of implications in terms of how we actually interact with these microbes, both in our body and out in the fields too. So yes, I will certainly, Vilma, I will certainly add you. Let me actually do this now before I forget. Put that there. Save that for later. All right, so I guess we got a few other questions here. Any recommendations for inoculants? So commercially speaking, there's a few inoculums out there. And the key with the inoculums, when you're purchasing inoculum from a commercial source, it's important to look at the number of propagules that are in on the label. So the higher the number of propagules, the better, or most likely the better the inoculum would be. Um, so so I would go with that. Look at those data, look at those numbers, and testing too. So if you're actually looking to inoculate your seeds with a broad spectrum, or you're looking to inoculate your legumes with nitrogen fixers, there's different ways to do this. So you could either do it yourself by just taking, let's say if you're looking for rhizobia, you can take some of those nodules and just make a slurry with the nodules, make sure that they're nice and pink. That indicates that you actually do have the rhizobia. If they're not pink to red, you don't have rhizobia. All you have is infection. And so you can take that rhizobia, make a water slurry, and then put that onto your seeds. That's an easy way to do that. I would recommend that over buying something, only because those microbes have been adapted to your own environment. So um, So that's one way of doing it. If you're looking at doing mycorrhiza, again, look at the propagules um, and I know this is not at the axis of many, but if you have a microscope, there's ways to actually identify whether or not your mycorrhiza inoculum did work. So it's just really ways to do these things yourself. I know that during our bulk order, we do offer some inoculums that are on there. Um, and obviously it's too late for this year. But next year there's a few that are listed there, like MycoApply and BioCoat Gold. I know that a few folks at NOFA do use those products. Um, so yeah, that's what I would recommend, you know. But again, Rodell Institute also has a really good way to how to cultivate your own mycorrhizae fungi from your own farm. And that information is out there. It's great. And again, I recommend using those inoculums with your own native microbes. And then let's see, another question here. Oh, sorry, go ahead.
I just wanted to say, like, for me, because obviously we are small here, it- I have found that for us it's better to put the inoculant on the soil
Hmm. Mhm.
that we're planting the seeds. That actually, like, I used to like grab a little bit of the, because it says, I think it says 4 ounces for 100 pounds or something of seeds. I
Mhm.
have never planted 100 pounds of seeds. That's like crazy. Um, but I think if you mix it with your potting soil, if you do in transplants, or if you mix it with the soil when you prepare, like I put it in the compost sometimes, or it works better. I think it has better results, at least for me. I don't know if others
Yeah, no, for
use
sure.
bioco
For sure.
cocoa.
And you know, and you can actually even take your own compost. You're making compost on site. You can take like a cold water extract and make a slurry out of that, and that could be your inoculum. You know, you have essentially managed your biologics that way, and that could also be used as an effective inoculum too. So compost could be used as that. And in fact, the way I view compost is an inoculum. I don't see it as a fertilizer. It's an inoculum if you're doing it the right way. Um, just whenever you're, you know, some of the things that may affect, again, if you go back to the slides too, is anything with uh, high, uh, drawing a blank here. Fertilizers with, you know, the use of fertilizers can also be detrimental to some of these microbes. And then so if you're doing both, you know, keep that in mind that if you are putting some of these high analysis fertilizers, you could be causing detrimental effects to those endophytes. Similarly, the use of nitrogen. If you add too much nitrogen to your soil, your plants are going to take that nitrogen instead of actually using the bacteria that would actually provide the nitrogen to the plant instead. So things to keep in mind. Um, cool. So I have a question here from Rosa. How does crop rotation affect the seed-soil relationship? Does growing varieties in different fields each season affect the rhizophagy cycle? Um, there's a lot of contention around crop rotation. Some people believe that if you actually have a high biological soil, meaning that you have great diversity in your soil, that uh, knowing what we know now and how plants get their nutrition, uh, crop rotation is only in effect from the conventional agriculture that comes from conventional agriculture because of what's being done to these endophytes. And so if you think about it, in a way where if you have again, high diversity, you don't necessarily need to be rotating your crops. However, that's not always the case. And so if you are suffering from some type of pest, again, it'll be important for you to actually try to get those your plant photosynthesizing to its max capability by either providing a biostimulant or foliars as well to make sure that that the plant's actually doing well. So, but now the question to your question about does growing varieties in different fields each season affect this rise of age cycle? That depends on, again, the soil. You know, if your soil is highly diverse, and depending on what's growing there too, depending on that seed bank that you have there, yes, this rise of age could be affected. You know, if you've applied a lot of fertilizer to this one specific spot, that could also affect that as well. Does that answer the question? Perfect.
Ruben,
Yes.
just a follow up on crop rotation. Um, I have a sense over the years that mustard family can be sort of antagonistic towards a lot of mycorrhizae. I don't know about other microbes. Um, and so it's something that, I've become careful about of, the um, things that I know are really reliant on that soil community, like tomatoes. I try to not um, have them too close to some of the stronger brassicas.
Hmm.
Do you have a sense on that dynamic? Are there other um, plants that can sort of antagonize?
Yeah. There's a that's a very good question actually. And so the I spoke briefly about that when I talked about the Phragmites, right? When you take the Phragmites and you take the endophytes out of that and use it as control for other weeds, right? So these same things are actually happening in your soil as you described right now, right? Where if you actually are growing, let's say a plant family, I can't think of any right now, but mustards are actually are a good um, are a good one there because you know anything in the Brassica family, they're just loaded with endophytes. And so these plants that are loaded with endophytes may actually have I don't want to say that they're fighting, but there's as you mentioned, antagonistic associations. Now, with brassicas, there's also um, even though they don't associate with mycorrhizae fungi, um, I believe it's actually actinomycetes that they actually relate to that are needed in order for them to actually create some of these associations. But again, this only comes from diversity in the soil. So if you know if you don't have those, then it'll be difficult for the plant to say, well, okay, I have these in here now. Um, yeah, that's a good one actually. I don't have a list of plants that would be antagonistic. There is a good book that I refer to called Plant Carrots Love Tomatoes. And it's a companion planting book. It was written a long time ago, um, but there's still a lot of good information on there in terms of uh, companion plants. But you know, after reading that book, then you start thinking, okay, well, are some of these companions actually um, are they allele chemical or are they endophytes that are actually being antagonistic? And that'll be a great point of research, right? You know, because for the longest time we thought, okay, well, um, black walnut, it's not going to let anything grow around there because of the was it thujan? Um, you know, is it the chemical or is it the endophytes actually that are saying, you know, like I don't want I'm gonna outcompete anything that grows around here. So I think there's still a lot to learn about these endophytes. Um, but yeah, that'll be interesting if we can actually get like a plant list, right? Of these endophytes are, not working here or there. I know that Dr. White is working with a lot of these folks on this topic. And so I hope to pick his brain at some point when we have more information. Right now, they're looking at plant families, and they're trying to essentially rewild some of these plants and bring some of these old varieties. Corn is a good one. They're taking some of this, this old corn. One of the things that I didn't discuss are, uh, like for example, the issue right now that we're having with Roundup Ready corn. Um, these plants, unfortunately, because of the way we're producing seeds, are not able to uh, communicate with the endophytes any longer. They lost the ability to do what they naturally do. And so there's this race now to try to go back and say, hey, we need to uh, bring this back by, breeding in some of these old varieties in, because these cultivars have lost their way. They don't know how to interact with endophytes any longer, which makes it sicker. We have to continuously apply more fertilizer. We have to continuously apply more pesticides. Um, and ultimately, we're consuming these plants. I don't think that's a good idea. Um, so yeah, so we have some issues with our plants and how they're actually interacting with endophytes. Um, and hopefully we can bring this all back. Um, Jesse, we use Korean natural farming preparations to inoculate soil and plant starts. We were thinking of adding mycorrhiza and azospirillum to the mix. Do you think it's necessary? Will it risk too much competition between the indigenous and store-bought microbes? Good question, Jesse. I don't know. You would probably have to do some trials side by side. With a lot of this information too that we're talking about tonight, the idea is for us to gather some of these principles and then actually go out into the real world, because all this information is actually coming from labs, it's coming from academia, and they're all well intended, but they're in very controlled environments. And it's up to us to actually take some of these principles and then apply them in our fields, gardens, whatever the case may be. Um, so uh, so it's important to actually do your own testing, right? Um, run trials, report back, let the community know what you're doing. There's no right or wrong way to do any of this. What may work for you may not work for the next person. So um, you should try it. But run some experiments. If you have access to a microscope, there's really easy ways to identify whether or not you do have these endophytes or an increase in endophytes, or if your inoculum is actually working or not, specifically when concerning the mycorrhiza too. All right, so Jamie, this is not super related to endophytes, but are there any risks or obvious benefits concerning soaking or drenching seeds in a fermented plant juice dilution? This is awesome because this is also related to, and when you say fermented plant juice, are you talking about like Korean natural farming? You take equal amounts of weight brown sugar to your plant and extract that? Is that what you're referring to? Yes. Okay. Yeah. So with fermented plant juice, that's an interesting one because there's a ton of hormones in there. There's a ton of other chemicals that are, you can't really know what's in there, but essentially you're getting the essence of that plant and then putting it into soil soak. Now, Korean natural farming also has a what's called the maintenance solution, which is what a combination of Oriental orbital nutrient. Um, I think it says an FPJ as well, and brown rice vinegar. And right now I'm actually conducting a few experiments myself where I took some clover seeds, inoculated them with different solutions, one of a Johnson Su, one of a uh, Korean natural farming, which is a soil soak solution. I also did a vermicast, some indigenous organisms that I captured with grass, actually, believe it or not. I captured these from grass clippings, which is totally possible. Um, and then what I did was I actually moved them onto some agarose plates, and then I used what's a DAB solution, which is like a dibenzo amine solution. Essentially, it'll color the bacteria brown, just to see, what would happen. And from all of the tests that I ran, the KNF appear to actually inhibit. First of all, it actually germinated much faster than anything else, and it also inhibited other bacteria from actually forming on the plate too. So obviously, that's just one trial with just one set of seeds. I didn't reproduce this, um, but these are things that everybody can be doing. It's not difficult. Agar agar is actually sold at health stores. Plates you can get online, and I use a pressure cooker to make the agarose. So um, again, these are all things that we can all be doing and sharing the information to. But yeah, give it a try. Certainly, again, Jamie, run some trials and see what works.
Awesome. Thanks. Sorry the camera's off. We're eating dinner.
No, no worries, no worries. Thanks for joining. Um, and Randy, yes, high phosphorus, absolutely. If you're using again high chemical analysis for phosphorus, that will certainly have issues establishing mycorrhizal symbiosis. Um, and along with that too, there has to be a slew of other microbes in order to facilitate this mycorrhizal inoculation too. So yeah, thanks for that. Yeah, and a podcast from John Kempf, which is awesome too. Great. Um, any other questions, concerns? Um, Emotional outbursts?
Ruben, can
Yes.
you hear me?
Yes.
Yeah, I was just going to say that you talked a little bit about the saving your own seeds, and the way that Dr. White was talking, that we really should try to do that where we can, because our saved seeds are going to have those microbes from our own terroir, if you will, which is really an important distinction. I think everybody who's saved garlic knows that. They've been growing the same garlic and gets better and better every year growing in their own soil. But most of us tend to buy seeds, and they're not used to our soils and our microbes, and they could be so much better if we try to save some of those.
Yeah, no, absolutely. If you look at all the slew of benefits that these endophytes have when they're adapted to your environment, because there's this thing, right? Adaptability. Um, you know, uh, Again, these practices will only benefit the farmer because um, well, not only the farmer, but in terms of like the socioeconomic impacts that this has too. Um, uh, you're growing plants that can achieve higher photosynthetic capabilities. Um, Again, it's adapted to your microbes, adapted to your soil, adapted to your climate, which is very important. We could even have microclimates that are different from, in one town to another. Um, so yeah, so there's a, and you're right, there's this thing of terroir, right? Where there's and actually there's a nice paper that came out on that too. I'll try to find it and send it to you guys. Um, that just speaks about that actually, and how when we talk about grapes grown in I don't know, California versus France versus certain regions, some of these endophytes are being attributed to these flavors that are present in here. So we're not, so and this gets into like the plant secondary metabolites in our foods, which we now know are responsible for the nutrient density in our foods too. So these are just benefits and um, and practices that, quite frankly, we should all try to be striving for. You know, and not only that, but also gives a sense of community too. When you're sharing seeds and saving these, there's a lot of stories behind that too. So, yeah, I apologize again. I'm not. I have a high fever right now, and trying to make it through here. Um, Anything else? So I actually have a few announcements actually. Let's see what is going on in the world of NOFA. So I would say we have about 20 plus events that we will be doing this year, both online and in person. So I urge you all to actually check out the website for events coming up. There's going to be some beginner farmer workshops that we're running. There's also a MDAR supported grant, which will do a series that's going to be at Dan Kittredge's farm. For those who don't know, Dan Kittredge is the executive director of the Bionutrient Food Association. And we have a nice series going on there with him, which will be really, really nice. And those are the ones I can think of right now. But yeah, we have a lot of fun stuff coming up. So I hope you guys can I can see you guys in any of these workshops. And again, the agroforestry call will be happening next month, and the following month of our soil health, which would be in April, we'll be covering compost teas. The idea of doing the compost tea online soil health call would be supplemental to a workshop that we're doing at the end of April on compost teas. So you'll get to learn how to make your own compost tea, how to evaluate it, and apply it as well. And yeah, that's all I got. And I'm gonna stop this recording.